Hello. I hope today is April 22nd, or if you're watching this video from the future and I look younger, awesome. I'm still using this video. Um, I'm gone. So you guys are finished up with your Unit 4 lab that you're studying photosynthesis or cellular respiration. And so I left you guys hopefully at the portion where you finished collecting all your data and you guys made your graph. Hopefully I helped you make your graph, change the title, change the axes, labels, making a key, making sure it's a proper scale. I should have helped you with all that. Most groups are pretty good to go. If you were gone when your group collected said data, you should be getting the spreadsheet and data from them. And you should be moving on to what I believe is the analysis questions. With that being said, make sure you link your spreadsheet within your lab. I need access to it, so please make sure to put the link of your spreadsheet in your lab. There's a portion of the lab that says, like, paste your spreadsheet link right here, URL right there. Beautiful. So please make sure you do that. With that being said, analysis calculations, okay? Before I left class, I had everyone type out in the analysis calculations how to calculate percent change for you guys' data. So I'm going to put the equation back up on the board in case you guys forgot, and I can do an example problem, okay? So rate of change. Okay, time to do the dab thing. There's cops coming by. I wonder. Okay, they left. They're not here for me. So rate of change. To do this calculation, you need your initial and final value. Okay, sending good vibes to those people. Okay, rate of change. You need your initial and final value. So the equation is you take your final value, final minus initial, divided by initial value. And if you want it as a percent, you need to, at the very end, multiply it by 100%. Okay? A calculator is going to be the best for this. Sorry, the glare of my overhead light. Over here, it says times by 100. So right here, it says times by 100%. Okay? So final minus initial divided by initial times by 100%. Okay? That's how you do a calculation to find if your value of CO2, ethanol, O2, whatever you're measuring, whether that value went up or down. Okay? So what's an example of this? Let's say I'm calculating amount of CO2, and let's say for my experiment I was doing different colors of light. Okay, so let's say I'm specifically looking at yellow light because most plants receive some form of yellow light from the sun, and this is my control group. And let's say I experimented with blue, green, red, and yellow is my normal. Okay, so let's say my final value. Actually, let's start with initial. My initial value for CO2 initial, let's say I had 500 parts per million, ppm. And let's say by the final value, let's say it was 400 CO2's parts per million. Okay? So as you can see, between my initial value over here, which is 500, I went down to 400. I need to know how much as a percent did I go down. So I'm going to use the rate of change equation. Okay? So I'm going to plug in those values for a rate of change equation. So I need to take final minus initial. So my final value is 400. So I'm going to take 400 minus my initial, which is 500. I got to do that calculation first. Okay? That's the first calculation. So if I were to plug that into a calculator, or you do it in your mind, 400 minus 500 will get you negative 100. Okay? So do that part first. Okay? That's what the parentheses are for. They're telling you to do that calculation first. Then I need to divide my initial. My initial value was 500. And at the very end, after you divide negative 100 by 500, you have to multiply it by 100% to find your rate of change for yellow light. Okay? And so if I do this math, I can pull out a calculator. I know what this is off the top of my head, but I will just show you me typing it in. Okay, sorry, my TikTok was going off. So, calculator. I have calculators in the classroom, just ask. They're in the green bins, or use your calculator on your Chromebook. There's no point bringing out your phone, and do not get hassled the sub saying, hey, I use my calculator on my phone. You're Josh, right? You have access to the World Wide Web. Use it. Find a calculator online. So 400 minus 500. 400 minus 500. Negative 100. Okay, that's the first step. Then I have to divide by 500. So I'm going to take negative 100, divide by 500. Okay, that's going to give me a decimal, which is negative 0.2. And then I have to multiply at the very end by 100 to make it a percent. So I got negative 20, so it's negative 20%. So that tells me that negative 20%. So that tells me my CO2 levels went down by 20%. That's what the negative's telling you. It went down. You have to ask yourself, what does that mean? Okay, you have to know what that means for the C.E.R. portion. So CO2 apparently is going away. It's being used up. And you have to ask yourself, okay, he's doing yellow light on a plant for photosynthesis. Is it doing photosynthesis or is it doing cellular respiration? Because plants can do both. Okay, so you have to ask yourself, okay, what's being used up? Well, it looks like CO2 is being used up. So then you ask, okay, which equation, cellular respiration, specifically aerobic, or photosynthesis, which one has CO2 being used up in the beginning of the equation? Okay, and if you remember correctly, CO2 is used up during photosynthesis. So I know photosynthesis is happening because CO2 levels are going down. Okay, so now I know, okay, with yellow light, the plant definitely does photosynthesis. Now when I do my other colors of light, I can compare it to this control group, okay, this negative 20%. I know that this does photosynthesis with yellow light. Now I wonder, if I do blue light, what if the percent goes up? What if the amount of CO2 actually goes up instead of down? Well, if it goes down, that tells me photosynthesis is happening. If it goes up, that's telling me maybe for the blue light, if CO2 goes up for blue light, that it's doing cellular respiration, okay? So you have to do this with every single control group and experimental group you guys did. Easier terms to put this in. Every time on your spreadsheet, you have a column, okay? A column of data that you collected, whether it was CO2 or O2, you have to find the rate of change for that column. So some of you guys may have tested a few different things. I had some groups testing different amounts of water for a plant. So some people did a column where it was 10 milliliters, 30 milliliters, 50 milliliters, okay? So you have to find the rate of change for each one of those values. Some people were doing amounts of light, whether you were doing 100% light, 50%, 0% light. So you got to find the rate of change for each of those, okay, for CO2 or oxygen or ethanol. Some people are changing the amount of sugar. Well, then you have to find the rate of change for your yeast, if you're testing yeast, for your brown sugar, powdered sugar, granulated, artificial, honey, whatever you added. Okay, so you have to do this for every single column of data that you have. And how do you find your initial and final value? You may be asking yourself. The initial value is going to be whatever the data was at 0 seconds, whatever the initial value was. And then if you collect the data for 180 seconds, well, your final value is going to be whatever data point was at the end of the column at 180. Okay, so that will be your initial and final for that specific experimental group or control group. Okay, so that's rate of change. You're going to need these calculations because they're going to be brought up in your C.E.R. With that being said, I'm going to erase this. We're going to go into the C.E.R. next because even though I made a document to help you guys with it, I'm assuming that some clarification is always a little nice to have. Okay? Hopefully, if I upload this on my personal YouTube channel, I may change the profile picture in the future, but right now it's a picture of me in front of a lake smiling with a backpack on when I was going hiking. So if I'm using that YouTube channel, if you look at the timeline of the video, I should be putting chapters, and so you can hover over the timeline and see, oh, when did he talk about this? Oh, this is the C.E.R. section. This is the part I need help on. I'm just going to skip to that portion. Okay. So on the lesson plan, I should have it somewhere. I actually have the lesson plan for when I'm gone on April 22nd. Again, I might use this video for the future. So if it's not the year 2025 for you guys, ignore this part. But there is a C.E.R. document I made that tells you what you guys need for every single sentence. Okay, so I'm going to try to go over those portions. So maybe I can make this chapter called this claim, the C portion of the C.E.R. Okay, so I have it where your homework is finish up 4.15 cellular energy experiment. You can use the following C.E.R. document, and it's hyperlinked, so you should probably click on it. And that should help you make your C.E.R. And I'm going to pop that up right in front of me right now on my other computer. So for your claim, it says you have to answer your experimental question. Your experimental question can be found if you scroll all the way up to the top of your lab. You should have your experimental question. Okay, your experimental question should be asking something along the lines of, for my example lab, I said, how does the color of light affect the amount of CO2 produced during photosynthesis for air plants? Okay, so your question, your experimental question is going to be at the top of your lab. You have to go find your experimental question at the very top, and you have to look at it, read the question, and you have to answer it finally. Okay, based on your data, how, at least for me, how did color affect photosynthesis for my plant? Or how did adding different types of sugar to yeast affect cellular respiration? How did the amount of light affect your cactus and its photosynthesis rates? How did the human's production of CO2 change if you were changing the person's different exercises? Okay, whatever your experimental question is, this is the moment we're going to answer it. So, first sentence of your C.E.R., you're finally going to answer your experimental question. So just flat out say, it seems that when you change the color of light for plants, blue and green seem to not be very good for plants. They don't do much photosynthesis, but it seems like yellow and red are fabulous for photosynthesis. Whatever your data tells you, okay? So you have to answer your experimental question. That's your first sentence of your C.E.R., okay? Whatever you're studying for your lab, you have to answer your question finally. First sentence. Second sentence is a lot easier. You have to answer, what is your independent and dependent variable for your lab? Scroll up to your, I believe it's your, it's not your procedure section. It is called your, it's just in the middle of your lab. I don't have a title to it. It just says, what is your independent variable? What is your dependent variable? Go look on your lab. It's in the middle of your lab. You're essentially copy and pasting it. The second sentence for your claim, I want you guys to just say it in a sentence. My independent variable is whatever you wrote in the box above for your independent variable. Okay, so whatever you changed, you're just going to copy and paste it, insert it down there. So for me, my independent variable was the different colors of light. And then you also have to say what your dependent variable is, so you also have to add in that sentence. My independent variable was the color of light, and my dependent variable was how much CO2 was produced, or whatever you guys are measuring, whether it's ethanol, CO2, or oxygen. Okay, so you're really just copying what's above your independent dependent variable below into the second sentence of your claim, and just copy and pasting it over. Okay, super easy step. Last but not least, the third sentence to your claim is you guys reflecting on your hypothesis. Your hypothesis was your educated guess of what's going to happen. Okay, my hypothesis, my educated guess of what was going to happen for changing the color of light was, I said, if you change the color of light, or if you change the color of light to something besides yellow, then the amount of CO2 produced is going to go down because yellow light is the best for plants to do photosynthesis. Okay, if, then, because. That was my educated guess. If your hypothesis isn't true, if your data shows that you were wrong and your guess was wrong, that's fine. Okay, that's fine. Being wrong in your hypothesis is completely okay. But you just have to state that you reject your hypothesis. If you are wrong, if your hypothesis, if your guess earlier was wrong, then you have to just state, given the data, the hypothesis for this experiment will be rejected, and yellow light was not the best light for photosynthesis. Okay, that's what I would say if I found out my hypothesis was incorrect. If you do have a correct hypothesis and you were right, and your data supports that you were right, you would say, based on the evidence, the hypothesis is supported, and whatever you said, whether you said sugar was the best for yeast, or humans produce the most CO2 during cardio activities, whatever you said, you're going to add that in there, okay? So based on the evidence, the hypothesis is supported by the data, and running causes more cellular respiration than any other exercise. Or you could say powdered sugar was the best sugar for yeast over brown, granulated, and honey, okay? So just accepting or rejecting your hypothesis is your third sentence. Sorry, getting a dry throat. I'm a little sick. Someone got me infected. And it's a week until my birthday, and I'm so upset about it. Okay. So that's your claim. First three sentences of your claim. Now we're going to go into evidence. And this is where your rate of change is going to come into play. So, evidence. First sentence of evidence. It says, state what the rate of change for your respiration or photosynthesis. So whether you're studying photosynthesis with plants or algae, or if you're studying cellular respiration, and you can study any creature for cellular respiration, you guys have to state what was your rate of change for your experimental groups. Okay. As a reminder, your experimental groups are the groups that you're trying to experiment with and see if anything is different. Okay. So for example, for me, I'm experimenting if red light, green light, and blue light was better for plants. So you guys calculated the rate of change from earlier that went into your calculation sections, and you should have gotten a certain percent. This is where you're going to report on those percentages, okay? So earlier I found that yellow light had a negative 20% for CO2 produced, which means CO2 went down, which means photosynthesis happened. So in my evidence, I would say for not yellow lights, these yellow lights, my control group, you guys are going to state your experimental groups right now. So I'll be reporting on my red, green, and blue light. So I would say for red light, the rate of change was negative 30%. For green light, the rate of change was positive 26%. And for blue lights, the rate of change was positive 15%. Okay, I just made up those values. But you're going to report on your rate of change that you calculated earlier in this sentence, okay, for your experimental groups, okay? So the ones that you're trying to mess with, see if these change anything. So report on those calculations there from your analysis calculations portion. Sentence number 2 is you're reporting on your control group. What was the rate of change for your control group? Your control group is your normal group. What does your plant normally get? Does it get a lot of water? If so, how much? If it gets light, what is the normal light it gets? It gets yellow light, okay, the light that's in this room. If it's yeast, what kind of sugar does it usually get? I usually give yeast granulated sugar. If you're testing different water plants in different waters, usually I give all my plants tap water, okay, so that's normal. So you're going to report on the rate of change for your control group, your normal group for the second sentence, okay? And that rate of change should be in your calculations portion. So you're just going to pull that percentage down. And you're going to say the rate of change for the control group, which was yellow light, was negative 20%. Second sentence is done. Nice job. Last but not least, third sentence, it says state qualitative data about your organisms during the experiment. So you're supposed to record quantitative, which is data with numbers. Okay, that's your spreadsheet. You have that. The sensor probably collected it for you. And you're supposed to also record qualitative data, which is data describing using words. Okay, so you're supposed to notice if anything happened that was unique. For example, if I changed the temperature of water for my fish, and that was the thing I was experimenting with, how did the fish's behavior change when it was in cold water versus its normal water versus warmer water? Did its behavior change? Because that's something worth recording. That's qualitative data, describing the quality of the fish. If you added yeast to brown sugar and water versus yeast to granulated sugar and water, did you get more bubbles? Bubbles are qualitative data. Do you see more fizz? Did you hear anything interesting? So you guys have to record qualities of your experiment, okay? If you were testing different fluids on a plant, for example, giving water to a plant versus sparkling water versus vinegar, did the plant react differently? Because for some of the experiments I saw when you guys add that vinegar, after like 10 to 15 minutes, that plant went from being super supported and stiff and upright and being like, hey, I'm happy. Once you got that vinegar, I saw its leaves start to sag a little bit as if it was starting to die, okay? That's describing the quality. That's your third sentence. Last but not least is your reasoning. 3 sentences. First sentence is restate if your independent variable had an effect on your rate of respiration or photosynthesis. So this is you essentially restating your answer to your experimental question, okay? So the first sentence of your claim, you answered your experimental question. This is almost doing the same thing, okay? Did changing the color of light truly affect photosynthesis for my plant? Okay, so this essentially asks, again, answering your experimental question, okay? And scientists like to have you repeat your answer multiple times in your conclusion, because if I'm reading a conclusion, it's 3 paragraphs. Often by the end of the 3rd paragraph, I forgot what you said. Like, what did you say was your answer? Did you say photosynthesis was going to be affected by the color of light? It's usually just a refresher, and it helps when I'm reading your lab that it refreshes me. So you're just restating essentially the first sentence of your claim and the first sentence of your reasoning. Number 2, it says explain why your independent variable did or did not have an effect on the rate of cellular respiration or photosynthesis. So you finally have to come up with a reason of why did the color affect photosynthesis for plants, or why did the different types of sugar affect the cellular respiration of yeast. You have to come up with a reason now. You can't just be like, oh yeah, it did affect it. Why? I have no idea. Gotta use that noggin. If you're really not sure, you can maybe research it. For example, why would I decrease the amount of light? I'm still producing or giving a plant light, but why is it when I decrease the amount of light, why did it switch over to cellular respiration? The plant. I'm still giving it light. Why did it switch over? It should still be doing photosynthesis. These are things you can research, okay, if you really are not sure. And last but not least, the 3rd sentence for your reasoning, sorry, I'm a little snotty. 3rd sentence for your reasoning is come up with 2 future studies that could be done with the same organism, same organism that you experimented with, okay? So if you experimented with yeast, you got to come up with 2 new experiments for yeast. If you experimented with an air plant, you got to come up with 2 new experiments for an air plant. If you did goldfish, 2 new experiments with a goldfish, okay? Has to be about your critter. So for example, if I study the different colors of light for an air plant, maybe I can do next time, I can propose doing a future study of maybe the different amounts of light. That's pretty good. Or maybe instead of different amounts of light for an air plant, maybe something I can also affect is maybe I could do different amounts of water I'm adding to the plant, okay? Or different types of water I'm adding to the plant. So you have to come up with 2 new ideas about your critter that you could experiment with in the future, okay? And I know at the end of your C.E.R, I'm going to have you put that. If you look at the very bottom of your lab, it does have a future studies section where it says place 2 future studies and blah, 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 aka what I'm explaining here. If you want, you can either put it at the end of your C.E.R, your 2 future studies, or you can put it in that future studies box. As long as I see it, I'm okay with it, okay? You can put it in either of the 2 spots. I just tend to like having the future studies in the C.E.R because a lot of kids will finish their C.E.R and then often not put anything in the future studies box and not realize they actually had to answer the future studies, like come up with 2 future studies. They often leave that blank. And so I'm having you guys add it to the end of your C.E.R, making a requirement in your C.E.R, because at least if you miss it and you don't put it in the future studies box, at least it's already in your C.E.R, so you're not going to lose any points. Okay, so I'm going to upload this video. I'm going to try to have it on the lesson plan for when I'm gone, so you guys can finish up the lab. Hopefully this helps. With that being said, there's probably an assignment that you have to do. It's probably, I don't know, it's probably called like 5.1. I'm going to look up what the assignment is called right now. Oh, no. It's called 4.14 Food Chain Energy. So you guys are just going to fill that worksheet. It's the classic assignment of, hey, these friends have opinions about food chains and where the energy goes between each level of a food chain. Do you agree with them? Yes or no? Explain why. Pretty easy assignment. So finish that up. Finish up your lab. I'm looking forward to grading them. Even though I'm gone, I'm going to try to grade them and keep them rolling in the grade book. Even though I'm probably going to be getting off a flight, and that sounds like it's going to suck. But I'll do it for you guys. So you guys, your grade is up to date even when I'm gone. Okay? So watch for the timestamps in the video. I should have probably said that in the beginning of the video, whatever. And once you guys finished up, make sure you press submit. Work on other assignments, okay? Don't try to dilly-dally on your phone. Sound good? Okay, lovely, awesome, epic, whatever. I'll see you guys when I see you guys. Good job on the lab. I was very proud of many of you when you finished it, at least in my 2025 year quarter 4. You guys did a great job, okay? Okay, awesome. See you soon.